Hello everyone and welcome to the NPTEL course in Herbal Drug Technology. In this particular week, we are going to understand and analyze different herbal products. Now, why do we need to understand and analyze herbal products? Herbal products are inevitable part of a global population usage, with as much as 80% of the global population relying on herbal remedies. Now, when this 80% of population relies on herbal medicines, there is always an exchange of knowledge happens. Now, some of these herbal remedies are well documented, some of them are still translated as folklore medicine, wherein they are just passed by word of mouth. So what happens is, when I try to replicate it, the similar effects are not observed, and for that reason I need a process called as standardization. In simple words, let's try to understand. Today you try making a tea or a coffee for yourself, and amazingly you found that it has turned out to be very well. The next day you try to make the same kind of tea and coffee, but somehow it's not matching to the quality or matching to the set notion that you had the previous day. What happened is, maybe the production technique, the preparation technique, the quantities of your tea changed, and thereby there was a whole change in the what are called as the organoleptic attributes of your product. Now, if I have to produce the same tea over and over again consistently with the same quality, I know step by step how it should be produced, for what time it should be kept, what vessel should be used, so much so that what milligram quantities or gram quantities of my tea, coffee powder, water as well as milk. Now, when I put a control on everything, that is what I mean by standardization. So standardization is a process to create a consistently same quality of product, and that is very essential for herbal plant material, because in my case, when I want to say a standardized herbal plant material is going to give me the same efficacy. If I don't standardize it, it will change on day-to-day basis, and then what happens is my efficacy suffers and my quality suffers, and hence standardization is a very vital quality control parameter, or standardization is a very vital parameter to assure the quality of your herbal products. Now, herbal products globally are known by different nomenclature. We'll just go through each of them, try to understand it, and then slowly through the next lecture onwards, we'll try understanding what are the methods of standardizing each one of them. So, starting with the herbal definitions, the first substances that we call, or first substances that we see, are called as herbal substances. So, herbal substances, when I say, are substances or products which are obtained from plant sources. Now, this can be just plucking it, taking it and drying it. Now, because they have been obtained from the plants, I can say that they are herbal substances. Now, this include plant parts such as your fruit, stem, roots, leaves, barks, etc., as well as some unorganized drugs. You remember, organized are the ones which are cellular in nature, whereas unorganized drugs refers to a non-cellular matter. Now, this include your juices, exudates, resins, volatile oils, or even certain what are called as unprocessed products such as your gums, mucilages. So, this includes your plant parts as well as untreated products such as exudates, gums, mucilages, or even resins which come out of plant and are kept untreated. So, if you just take it as such, say for example Vasaka leaf, I am consuming it as such, or I see it in markets as such, I will call it as herbal substance. Not only that, the same your dink, that is your acacia, as well as your opium, latex of it is also classified as herbal product. The next category is herbal preparations. When I take this herbal substances and process it, now this processes can involve extraction, that is I boil it in certain solvents, distillation especially for volatile oils, fractionation to purify it further, even fermentation to prepare asavas, aristas and Ayurvedic preparations, even your tinctures or even processed exudates, not the native exudates but after some processing they are called as herbal preparations. Example of this include your essential oil like your jatamansi oil or your papaya leaf extract which is given for the treatment of dengue. Now moving on to the next definition is a whole product which comes out of it. A medication which is sold OTC or on prescription which is obtained from herbal sources is called as herbal medicine or herbal medicinal product. So herbal medicinal product or herbal medicine is a finished, nicely labeled product which contain active. The active can definitely be a herbal product which include your plant parts or combinations or even herbal preparations such as oils or certain processed extracts. This can include your unorganized material like your juices, gums, oils, mucilages, and with that it also contains excipients. So these are formulations which are ready to use and in certain cases will require blending. So definitely in like your tablets and capsules they will also have your excipients which are added which have been approved by the pharmacopoeias. So example of this finished products include your Triphala churna or even ashwagandha powder or include your laxative tablets such as your Senna tablets. Now once you understand these products, we know that depending upon the source, depending upon how they are processed, with a simple imagination of your tea, a slight change in any processing condition, processing temperature, processing quantity is going to change the quality of my tea. Similarly, that is going to change the quality of my medication and hence I need to do the standardization. So what exactly is standardization in herbal perspective? So in herbal perspective, the American Herbal Pharmacopoeia defines standardization by a very meaningful sentence. It says it's a body of information and control that is necessary to ensure that you get a material of a uniform consistency. So whatever you need information, whatever you need in terms of quality control, you should have it as a standardization parameters. That too will help you ensure that your tea or that your product is always uniform. So it's a process of establishing uniform guidelines. It's a process of specifying giving a particular specifications for your products in terms of its identity, in terms of its composition, consistency and definitely efficacy. So this specifications will ensure that all of these parameters remain the same. It also in addition to that ensures that because it is now been proven it is no longer containing any contaminants and as a result when I just take 1 gram of this, it is going to accurately give me the same efficacy as my previous medication. So that will definitely give dose accuracy, which is little difficult in case of herbal medicine as there are a lot of geographical, regional and other variations observed despite uniform processing conditions. So just to start with, if I have to set a standard, what are the standards that I set with? I have to compare it with something. Imagine the case of your chai. When you had your best chai, now you have a notion that my best chai tastes like this. So what is the notion of your herb? So if this is a genuine herb, it has to be something like this, and that is when I say botanical reference standard. So botanical reference standards are well standardized botanical reference material. Say for example in the picture here you can see calendula, flowering heads. So this is a botanical reference standard. Why? You can see it's DNA barcoded. What does that mean? A complete DNA identification of that material has been done, and it is sure from DNA identification that whatever is there with you is Calendula officinalis. Not only that, there are physical parameters, chemical parameters, not only that, molecular characteristics. So you can do some physical chemical test to ensure that botanically also. See, what happens is my clove can be clove by DNA, but what if I sell you clove which is devoid of oil? The plant material will be genetically the same, but I have already removed the oil, so it is chemically incompatible but DNA wise matching. So I have to see physical parameters, chemical parameters, molecular parameters along with the DNA barcoding. So botanical reference standards is that set of plant material which you can compare with to see if your raw material is of the set standard. So you can use that as a comparative standard. In a similar manner, we also have what is called as phytochemical reference standard. So you see that in your market you have numerous haldika samples or turmeric samples. Now this turmeric samples, you know, you have curcuma longa, curcuma amada, you have different curcuma species varying in their content of yellowness. So this yellowness is due to set of yellow pigments which are called as curcuminoids, and the major pigment in that is your curcumin 1, which is shown in the figure here. Okay. Now this figure is of curcumin. Sometimes it shows a diketo structure or sometimes it isomerizes into a ketoenol form. So this curcumin, what it does is I can use this as a phytochemical reference standard. So this phytochemical has to be there in all my curcuma or haldi species. Now depending upon which species I take, my curcumin content is going to vary. But I can say for curcuma longa, my curcumin content has to be 6%. For my curcuma amada, my curcumin content has to be 5% or even a range. Now if my samples fall in that range, what does that mean? That means that my curcuma is of certain quality. If it is following below the range, that means it has not been cultivated well, or due to some conditions my curcumin content has gone down. Now this could be even the plant disease which can decrease my curcumin content. But when my curcumin content goes low, what happens is it is not going to be efficacious. I may consume 1 gram of haldi powder, but that is not going to give me efficacy because the curcumin in that haldi powder is less. So phytochemical reference standards are phytochemicals of known purity, and with that or using that you can see how much amount of phytochemical is there in your drug material, your herbal material, your formulations or even herbal medicinal product. So once you can use that or once you compare this, you know that how much is the curcumin content of your plant, how much is the curcumin content of your formulation, and based on that you are going to know that it's going to be therapeutically efficacious, is it going to be safe, is it going to be consistent. So during monsoon what is going to happen is if the curcumin content is going 5% and during the summer if the curcumin content is going 8%, I have to change my dose to bring the same curcumin content, and that's why your phytochemical reference standards are important, and most of your herbal material as well as herbal medicinal products which are there in market are standardized generally by phytochemical reference standards. Now these standards are something which can help you in quality control, it can help you in ensuring your consistency, reliability as well as in product development. So imagine during some processes curcumin is getting lost. I am extracting it in a solvent in which my curcumin is not soluble. So I will be able to analyze and see that in my final product is the curcumin going to come or not. So definitely, so this markers help me estimate. Now this standards are also important for identification. Imagine somebody is trying to sell me some other yellow color root, calling it as curcuma, When I do a simple test called as thin layer chromatography with curcumin, this is the curcumin standard, and I see that there is a spot matching at the same RF. I know definitely that yellow colored drug has curcuminoids. But imagine if this is not matching, that yellow color is going somewhere else, that means that drug has some other yellow color which can be artificial, or some other yellow pigments which are not curcumin. So then I can come to know that whether the raw material is genuine or not. That is another application of your phytochemical reference standards. Now, there are books which often help us in identification. How do we know? So, these books include your pharmacopoeias. Every country tries to develop its own pharmacopoeias to set up standards, or to set up minimum standards that your raw material should be of in order to be compliant or of good quality. In addition to that, for those countries which do not have their own pharmacopoeia, the World Health Organization (WHO) has developed its own WHO monograph and quality control methods for that. In India, we have Indian Council of Medicinal Research, which also has developed numerous monographs, almost to huge volumes, 16-18 volumes of medicinal plants, so that we are able to do quality control and ensure consistency of herbal raw material, which can then be reflected in the herbal product. Now, coming to how it is done, there are two types of methods which can help in standardization, and that is what we do as quality control methods. So, quality control methods we can broadly classify as quantitative methods as well as qualitative methods. The quantitative methods are more of yes-no method. Say for example, you give me a yellow herb or a yellow powder and say it's a haldi powder. I will run a TLC of that haldi powder like we just saw. Is the spot matching? Yes, then it is haldi. Is the spot matching? No, then it is some other powder. Meanwhile, say for example, there is a drug which is rich in tannin. I can do a simple test for ferric chloride. So, this is a drug extract. I can do the extract in water and I just add a drop of ferric chloride to it. If it is giving me green color, I know that the drug is rich in condensed tannins. Suppose I get a blue color, I know that drugs is rich in hydrolyzable tannin. So, say for example, amla. Amla is something which is rich in hydrolyzable tannins, and if I treat my amla extract with ferric chloride, I should get something like this, a bluish coloration. Instead of blue, if I am getting a green coloration, that means it's not amla, it's some other herb. Similarly, I can just do a simple microscopic test of that amla. This is what I call it as transverse section. So, I cut my amla and put it under microscope, or cut my plant and put it under microscope. I will be able to identify the plant part, the botanical arrangements, and basis that I will come to know whether it's amla or not. So, qualitative tests are more of a yes-no test, which will tell me what is called as, is the drug present or no, is the drug genuine or no. So, I will just write it as yes, no kind of test. When I come to quantitative tests are actually the tests that will tell me how much. Say for example, my haldi, definitely it matched the spot. So, it had curcumin, but how much curcumin does it have? I have to do some tests, quantitative tests, to quantify the amount of curcumin. So, all those tests which help me tell how much of the compound is present, they are classified as quantitative tests. They are more to do with numbers. So, you will often get some reading. So, it is like a 6%, this much volume, this much absorbance. So, I can do a UV spectroscopy quantitatively, I can do an HPLC, or I can check some physical methods, or I can check some microscopic methods to see the values, such as your lycopodium spore method, or even your leaf constants, which will tell me, this much stomata are there per millimeter square, or this much vein islets or vein terminations are there per mm square. So quite likely your drug is what you call it as a genuine herb. So qualitative tests are a yes no test, quantitative tests are more of a how much test. Now if you see WHO guidelines, WHO guidelines also categorizes methods for standardization. So it gives numerous quality control methods for standardization of herbal drugs, and these methods can be divided into numerous categories. These categories include organoleptic, something that we can feel with your five senses. You say for example, you've been told by your parents to purchase oranges from the market. See, you don't go with a definition, you go with a notion of orange. So you will handle or you will see a fruit which is orange in color, you will see its texture which is kind of little rough and spotted. Then you will take the aroma, it's more of citrusy and sweet, lemon and citral like. Then you will feel it. So you will see it, you will smell it, you will feel it. You might even want to taste a sample of it. All of this comes through your organoleptic evaluation. But yes, in herbal drugs, not everything can be tasted, but you can definitely touch, feel, see different drugs. Then you can do what is called as macroscopic analysis. Now this macroscopic analysis helps you in identification of drug macroscopically. Say for example, I have to differentiate between drumstick leaves and kadhi patta leaves. I know macroscopically that kadhi patta leaves, typically have what is called as the very acuminate apex, whereas my drumstick leaves are your compound leaflets. So I should see compound leaflets when I am going to drumstick leaves, whereas for kadhi patta or curry leaves. So this macroscopic evaluation, I need not put it under microscope, just plain evaluation, the texture, all of this, or just visually seeing it, I can come to know. So that visual inspection is more often referred to as macroscopic. So for your cinnamon barks, those quills, or certain times even for your attas, the color of the atta can tell me. Now when macroscopy is little limited, we can even go to what is called as microscopic method. Now you take the same samples or specimens and put it under microscope. You might want to do a transverse section, you might want to grind it and convert it into powder. You see different organelles. Now these organelles include your tissues, parenchyma, collenchyma. These organelles include your ergastic cell contents like your starches, calcium oxalates, the arrangement of tissues, monocots, dicots. The physical parameters are also something that you can see. So in physical evaluation, you have moisture content, you have swelling, if a drug swells, or you can have for oils, refractive index, you can have for oils viscosity. Not only that, you can even see the chemical parameters, what all chemical classes it contains, and in the end to see the efficacy, we also have biological methods. So WHO gives different ways or different methods to assure the standardization. This includes organoleptic, microscopic, macroscopic, physical, chemical as well as biological methods of evaluation. In the subsequent session, we will be seeing each of this in much detail. So here are two examples of standardization. So say for example, when you go to a shop, you might come across some products like this. Say for example, this is what is called as ashwagandha. So ashwagandha, in case of this, are what are called as capsules. So in ashwagandha capsules, what they say is it is containing 7% vithanolides, that is what you say when you say standardization. It contains 1% alkaloids, so 7% vithanolides, 1% alkaloids, and specifically phytochemical reference standard, it has what is called as 0.25% vidaferin A. So when I am having this capsule, I can be assured that in each capsule, if it is a 100 milligram capsule, at least 7% are vithanolides, 1% I have a alkaloidal content, and in that 0.25% I have vidaferin. Similarly for Brahmi, if I am taking Brahmi supplements for my brain or from my memory, I have to see that I get loose Brahmi powder versus standardized extract. So here I have standardized extract which is clearly telling me that each of them have at least 20% bacoside. Now bacoside, your Brahmi has a mixture of macoside A, B, C and so on. So it is the total bacoside content, but I can be assured that in 100 milligram capsule, 20%, that is 20 milligram is going to be your bacosides. So with that ratio, I am really good that I understand how much drug I am taking and how much efficacy is there. But if it is a just unstandardized powder, it is going to vary from season to season, it is going to vary from manufacturer to manufacturer. So standardized extracts are much reproducible and consistent, and that's the very need for standardization. So here are a few references if you wish to know more about standardization and standardized extracts, and thank you everyone for your patient listening.